All right, so good morning, everyone, and welcome to our biology departmental seminar. Today's speaker, of course, is Nancy Shackell. Nancy is a senior research scientist at BIO. She also is an adjunct professor here at Dalhousie. She received her Bachelor of Science from McGill University, and then she did a PhD with Roger Doyle at Dalhousie. Actually together with the late Ransom A. Myers, and this is my little plug for a lecture we're holding tonight. Please log on at 7 p.m. To our Ransom A. Myers lecture in Science and Society. Our speaker today is Daniela Fernandez, who's the founder of Sustainable Ocean Alliance. She talks about youth and the sea, building the next generation of ocean allies. I would ask everyone to consider this lecture, which will be, I think, very enlightening and interesting. Also a celebration of Ransom Myers' life. So back to Nancy. Nancy's research on the effects of fishing, climate change, and other factors on marine biodiversity is in support of DFO's core mandate to promote sustainable fisheries and conserve marine ecosystems. She had a number of functions in the institution. For example, she co-led the Aquatic Climate Change Adaptation Services Program in the Atlantic region. She's also a co-chair of the US-Canadian Collaborative Framework together with NOAA for studying fisheries vulnerability to climate change. So clearly a lot of expertise that's highly sought after on the effects of climate change. And her most recent appointment that I didn't actually know about was she was appointed by the United Nations Director General as one of only 15 scientists from around the world to draft the 2023 Global Sustainability Report in support of the 2030 UN Sustainability Agenda, informing the 17 Sustainable Development Goals and their implementation. And she's the only marine scientist on that panel. So in just chatting before this lecture, I was hoping that we could have Nancy again at some point to talk about that experience, which I'm sure is a fascinating and insightful one. So Nancy, thanks again for joining our departmental seminar, and over to you.
Okay, thank you very much, Boris, and welcome everybody. I think everybody's had enough time to read the title slide and all of the authors that are involved. So the origin of this is what I'm presenting today is actually a review in fisheries science and aquaculture. And all of these researchers stem from various parts of Atlantic Canada and one from Alaska. And we were all working semi-independently. And before COVID, we were going to meet with Daniel Hennon in the US, and then COVID struck. So instead, we put our minds together and produced this review. And today I'm going to tell you about it. So the outline of this talk is, first I'll describe Atlantic halibut, its distribution and current status, and then go on to talk about the current management structure. Then I'm going to take a minute to focus on halibut around the Hague Line, which is a focus on the US-Canada border. The Hague Line divides the US and Canada in the Gulf of Maine. Then I'm going to set the stage in terms of halibut habitat and warming in the Northwest Atlantic. What's happened as a result of warming? And then finally, I'm going to bring everybody's research together and report on the flurry of research within the last decade on genetics, migration and spawning migration, juvenile nursery habitat, life history traits, and then I'll present a near synthesis because we, of course, have many, many more questions, but we believe that where we are now will help us to manage sustainably. Nelden Hare is going to be the key person, Brad Hubley and Lingwell Lee, to sort of bring this into the stock assessment process. So Atlantic halibut, you can see up here at the top, I have to get my laser pointer. Okay, up here at the top, we can see in the dark red, this is where Atlantic halibut are distributed all across here, but they're never really found south of 40 degrees north. So they really are a cold water, large flatfish. They're long-lived, late maturing, and can grow up to 3 meters. The females mature at a larger size than males and are also larger size at age than males. And it is thought, well, we know that they batch spawn on continental slopes from January to early spring. And in some instances, we know where that is. And in other instances, we don't. We have incomplete knowledge about where they spawn. We believe that eggs are suspended at midwater depths for around 13 to 20 days. The larvae are bathypelagic for about 90 days before settling in nursery habitats. And these fish, being a large fish, they can actually travel long distances. So what's happened in the last 2 decades? Well, we can see here we've got landings both in Canada and the US since 1950. We can see that the landings fell in Canada, they rose and then they fell again. And then in the last 2 decades, they have been increasing. And not only that, they have been increasing in value as well. So here we've got the value of landings for the Atlantic halibut compared to the Pacific halibut. And you can see that we start to rival the value of Pacific halibut around 2010 and even surpass that in 2019. So Atlantic halibut is currently the second most valuable groundfish in Canada, despite accounting for only 7% of the volume of Atlantic groundfish landings. So it has become quite important in our region. Not as important as lobster, but important. So where are they? Here we've got the distribution of Atlantic halibut, and these are just IDWs from surveys, from research vessel surveys, which largely catch juveniles from 1990 to 2013. So wherever you see red, there's more than an 81% chance of finding halibut. And you can see that the main concentration is really on the Scotian shelf, but we see also concentrations throughout the Gulf of St. Lawrence and along Newfoundland. These are the Grand Banks. So how do we manage these? We manage them principally as 3 main stocks. So here we've got the largest stock, this outlined in blue, almost looks like a little bit like an alligator or some kind of creature. And that is the Scotian Shelf Southern Grand Banks, or NAFO divisions, 3NOPS, 4VWX, 5ZZ. And these NAFO divisions are what we traditionally either use to analyze stocks as a unit stock and or collect statistics for landings. So right now, this stock is assumed to be one stock stretching from southern Newfoundland all the way down to southwest Nova, following my cursor. The second largest stock is in the Gulf of St. Lawrence, and this is NAFO divisions 4RST, outlined in amber gold, and you can see that here. And then the Gulf of Maine and Georges Bank stock in 5YZ right here. So it would be the smallest of the two. But what you can also see is we follow the exclusive economic zone that defines where countries can fish. We can fish inside this exclusive economic zone and the US inside this exclusive economic zone. You can see that there's a border right here. So this animal is distributed across international borders, both in the US and Canada, but also out into high seas. So we're going to focus now on the Hague line, which is this line following my cursor. And we'll go right here. So on one side, we have the USA, and then on the other, we have Canada, and then we have the Hague line just dividing this region. And what you can see right away is this is the Gulf of Maine. So it looks like sort of one ecosystem or a large scale ecosystem, and then Georges Bank, which is also dissected. So we've got this artificial, well, from an ecological perspective, an artificial border sort of cleaving through the Gulf of Maine and the Georges Bank region. So what does that mean for halibut? For halibut, we can see that on one side, halibut is actually MSC certified, so Marine Stewardship Council certified, and that was done in 2013. And that really means that we can safely eat that fish because it is sustainably managed under the criteria of the Marine Stewardship Council. In contrast, we have on just across this line, Atlantic halibut is actually considered a species of concern by the NOAA National Marine Services. So what we have here is the halibut was identified as a species of concern. It's quite a contrast. So we decided to look into that. And some people thought maybe it was climate change. So we looked into that by taking a whole bunch of surveys, or the same survey, 6 surveys that sampled both sides of the border simultaneously. So one survey went up into Southwest Nova, this area here, and also sampled down here. And we took those 6 surveys from 1965 to 2015, and this is just a snapshot of all of them together. And right away, if you see where my cursor is and squint your eyes a bit, you can see that the majority of halibut are on the Canadian side. And we can also see that in this graph, where we have proportion of sets occupied by year, and Canada is in red, and the USA is in blue. So you can see Canada, it always has greater occupancy, and on average 5 times more fish from an occupancy perspective than does the US, although you see a little rise here. We also looked into maybe they were habitat limited, but when we looked at that, habitat was not limited in the US. So it wasn't habitat that was limiting similar occupancy and or recovery in the USA. We also know that bottom temperature was not warming through most of the time series. It has in recent decades, but certainly wasn't in the 60s and 70s. So what accounts for the persistent difference? So we can go back in time for that. And here we have Nell Denhare's favorite slide. And this slide is a snapshot of many people's research into the history of halibut fishing in the Gulf of Maine. So in terms of fishing history, in the 1700s to the early 1800s, halibut was actually considered a nuisance in the US cod fishery because it didn't taste good salted, not as good as cod was salted. But then as time went on, the access to ice became easier in the early 1800s, which meant that halibut were more desirable. And so in response to that, there was a commercial fishery that was opened in 1836 in Massachusetts Bay, and then it was quickly fished out. It was fished out there, and so the boats moved on to, this is Nantucket Shoals, and onto Georges Bank. So, and both these areas were overfished by 1850. Then by 1880, it was really overfished. So I guess there are degrees of overfishing. And in 1880, they declared a collapse. But under the Treaty of Paris, we didn't have an exclusive economic zone yet. So under the Treaty of Paris, the US fishermen went into Southwest Nova, and Canadians actually didn't get into the fishery in a serious way until the 1900s. So standing back, what you can see is that there's almost serial spatial depletion of different fish, but not necessarily in Canada. Well, they were depleted in Canada, but not to the great extent. There's also this eastern Gulf of Maine area that was not so heavily fished. The majority of fishing was concentrated in Massachusetts Bay, coming down into Nantucket Shoals and Georges Bank. So that's one piece of history. So we thought in discussions among ourselves that maybe the US halibut were overfished and never fully recovered, as they're so long-lived and they operate on smaller spatial scales than assumed. And this sort of accords with something that's going to come up again and again, and I introduce it here. And this is something called contingents. Contingents are groups of animals who can be part of a population, a subpopulation, but they actually just work together. And what can happen in terms of contingents is that you can have a breakdown of contingents if some are overfished and or that there's a breakdown in socially transmitted traditions. I also have here the halibut fishing grounds in 1929. This is a picture from the fishing grounds of the Gulf of Maine by Walter H. Rich. And you can see the patchiness here and where they're still fishing in 1929 within the US. And line, that previous map, was already an 80% decline in the landings since 1895, and also the previous places hadn't, the Massachusetts Bay hadn't necessarily refilled. So what we've got here is the landings from 1893 to 2005. And you can see now in more recent years that at least up until 2005 that the landings have gone way down. So we'll go back. Following my cursor again, we've got 1965 to 1978, 79 to 92, 93 to 2005, and 2006 to 2017. And what I want you to notice here is that the patches in this area, United States and Canada, start to fill in and indeed darken so that there are more, the density is higher, the occupancy is greater, and they seem to be increasing since 2006. So we asked ourselves, is this true across the region? Well, it is. Here's an example of the plot of the mean number of Atlantic halibut per tow for the DFO summer ecosystem survey sets since 1970. So these are considered juveniles. And what you can see is the solid black horizontal line is the long-term mean from 1970 to 2019. That since around 2008, 2008, it has been above average, and indeed almost sometimes twice above average until just lately. And we asked the question, well, why? We did have some background knowledge. We knew that bottom temperatures in our area had started increasing about 2005. And also climate change is pervasive. So we focused on climate change being one of the usual suspects. So we asked the question, how do climate change and habitat availability factor into the expansion of Atlantic halibut in Atlantic Canada? This is work done by Andrew Chick. It's in prep. And what I have here is the climatology of a high resolution or ocean North Atlantic model, also known as BNAM, from 1990 to 2018. This model gives us the bottom temperature for any given year. And we can actually, it's usually used for climate projections, but we can also use it to examine annual and seasonal hindcasts. So that's what we did. We used it to examine the change in area of halibut thermal habitat, 3 to 17 degrees, as well as the growing degree days, which is just the length of the growing season as we get warmer and warmer. So really, we're just asking how much of that habitat between this window changes over time. And we cursor is, I have this is a map with all the NAFO divisions. And then corresponding in these two graphs, we've got percent thermal habitat and growing degree days in an earlier, from 1990 to 2003. So that's like the sort of reference period. And then we have the percent change from 2004 to 2018. So as an example, if we look at 3N, it didn't really have a lot of thermal habitat to begin with in 1990 to 2003, but it's increased more than 50% since 2004, in the 2004 to 2018 period. And you can see that's true of many areas, mostly in the north. The far north has not increased, but those areas in the south that already had a lot of habitat, so to speak, some have increased a bit and some have not. We get the same general pattern for growing degree days. So on average, the majority of this area has increased in both thermal habitat as well as length of the growing season. And from this, we know that it's only part of the story. We did not look at other factors such as competition from groundfish or seals, but we know that climate change will be a persistent part of that story. It will not go anytime soon. So we wanted to take a chance to revisit the spatial stock structure in an era of climate change. you know, when there is no climate change, we can afford a lot of assumptions. We can afford that things will stay stable in a static system in terms of a stock assessment process. So a stock assessment in a less dynamic world can afford all those assumptions, but under climate change, there's an impetus for basic ecological research. And that is like here's my push, the big push to understand the mechanisms so that response to changes in drivers can be anticipated. In this instance, it will be the response to temperature changes. And then I put good stuff for managing in a dynamic environment, because the stuff encompasses all of the information that we have learned. So as a review, before we sort of revisit that spatial stock structure, a unit stock can be considered, a unit stock assumes that individual level properties, growth, maturation, reproduction, and survival schedules, that those are uniform across the domain. So every fish grows at the same rate, every fish matures at the same size or at the same age, et cetera, et cetera. Those get translated or feed into population level properties, which then get into fishery level properties, which tell us how much can be fished sustainably. So it's really crucial, the assumptions that we put into a stock assessment in order to allocate or provide advice about how much can be fished sustainably. So a unit stock can exist across the seascape as long as these properties are spatially uniform. So we went about, and I'm now going to provide you on updates on the Atlantic halibut spatial stock structure. All of these genomics, migration, spawning areas, larval, drift and retention, nursery areas, growth and maturities, all of these inform us about the spatial stock structure, or even spatial structure per se. And I'll just say right away, we have a little question mark on the larval drift and retention. We've only ever found 5 larvae, not we, everybody from the Gulf of Maine up have found larvae to even know where they drift or are retained. So I'll first start with genomics, and this is work done or led by Tony Kess and Ian Bradbury, DFO. And what they did is they used 86,000 SNPs from 734 samples using a newly sequenced reference genome. And what they did is they found 3 main sources of genomic variation: a potential chromosome rearrangement, a sex-associated region that separated males and females and unknowns, and then also a subtle but meaningful genetic distinction between the Gulf of Maine, where I have my cursor, and the Scotian Shelf Grand Banks halibut, but no distinction between the Scotian Shelf Grand Banks and the US. So we look at the dots, you can see mostly purple up here, and that would correspond to the Gulf of St. Lawrence. They do separate, but you see some smattering. So it's a subtle genetic difference, but it's good enough to confirm the current management division between the Gulf of St. Lawrence and the Scotian Shelf Grand Banks. And they believe that is associated with environmental variation. But spatial structure can still matter in the absence of genetic differences. So genetically homogeneous group of fish might be more than one phenotypic stock if they exhibit phenotypic or behavioral differences. And that matters because in terms of phenotypic differences, that can arise from variation in life history parameters. For example, growth, survival, age at maturation, and fecundity. And there's a great example actually from Pacific halibut. They're genetically homogeneous throughout large parts of its range, yet they demonstrate different, oops, I went too fast, different life history parameters on a geographic gradient. And they have to take that into account when they're doing their stock assessment, because it affects the assumption of uniform life history trait parameters in the stock assessment population model. So we can have similar a genetically homogeneous group, we still have to worry about phenotypic differences. And then we also have to worry about behavioral isolation and segregation, which can occur over timescales that are too short to derive genetic divergent, but can also form stock structure. And that requires more complex management strategies as well. If we ignore all of that, there is the possibility that we could undergo or have serial depletion of contingents or subpopulations that can occur due to differential fishing effort across the seascape. And here's what I'm going to use this slide from a teaching class, lecture 13, to demonstrate that. Here we've got metapopulation. Metapopulations are spatially structured populations. So on the x-axis, we have the index of connectivity from populations that are highly isolated to those that are highly connected. And then along the y-axis, we have the variation in patch size. So they're mostly small to highly variable. And what we can see is if we can look, we can have any series of types of metapopulations that stock assessment or allocating fishing we would have to worry about. And I'm going to draw your attention to this one here, where we have occupied patches and we've got empty patches. Well, imagine if where my cursor is, we were to overfish this one, we can see that we are leading to more isolation, or we're cutting a connection, which can lead to highly isolated small populations. And the reason I took time to do this is actually what happened to northern cod. So we've got a precedent. So we've got halibut now doing really well. And we also know what happened to northern cod. That was studied quite a bit. And here's sort of an array of papers that looked at the spatial structure and metapopulation and found that cod indeed were serially depleted. So we'd fish in one area and the fish would run out. We'd move to a next area and those fish would run out, and we're decreasing the connectivity between any remaining areas or even the rescue effect. So if core high density areas are targeted by fishing boats, you can see that these subpopulations can easily be overfished. And I've also put contingents here. So groups of fish can easily be overfished. This leads to local extinction, which eventually leads to widespread extinction. And so the bottom line is that can happen either through differential fishing pressure and weak connectivity or different population growth rates. So there we are. Here we are on the left-hand side in the 1960s with cod. They had more than a blackboard, but it serves to illustrate the point that back then, when we were studying cod, we did not know everything. We've learned so much from cod, and here we are, starting around the 2005. Now we're thinking about halibut. We've got pop-off satellite tags, data storage tags, genomics, better statistical approaches, and we have evidence of serial depletion on contingents and subpopulations, and we also have evidence of local adaptation. So in my view, we are faster, stronger, wiser, which allows us to be more proactive. So let's go in that direction. And here I'm going to tell you, here I have put the Atlantic halibut landings. So these are where halibut are caught in the halibut fishery. For those of you who are familiar, this is known as the Gully along the shelf in Southwest Nova, also up in the Gulf of St. Lawrence. This is where Atlantic halibut are targeted. And if we compare it to where juvenile halibut are found, we can see that generally there is an overlap. So given our history, given what we've learned, knowing that the fishing industry will go where the fish is, we can use that information to better manage the fish. And so that was true from 2014 to 2018. These are the Atlantic halibut landings, and this is the distribution. And interestingly enough, it was also true from a paper from 1958. This is now the percentage of halibut landings taken in 1953-54 from each subdivision. And I'll draw your line to these hatched regions. They were fishing where fish are currently concentrated now, back in 1953-54. So the fishing overlap with juveniles. Two things it tells us is the fishing industry know how to fish very well, and the halibut distribution has been persistent. So the next stop, we're going to now look at what we know about migration and spawning areas. So this is a whole slew of research that is coming out of Newfoundland. Some has been done on the Scotian Shelf, and I think Dalhousie OFI might also be responsible for another project on the Scotian Shelf. This is work coming out of the Gulf of St. Lawrence and Maine. And here you've got Arnaud Lebris, who's double tagging a halibut near Port au Choix. And then I put in Andrew Seitz in the Pacific. He's also an author, but a huge fan of Atlantic halibut as well as Pacific halibut. And the comparisons are very, very interesting. I'm going to tell you the punchline first, and then I'm going to walk through how we got to this punchline. And the punchline or perspective is that halibut conform to the contingent groups theory. The contingent theory is that contingents or groups within a population develop migration pathways as a result of environmental cues at early or different life stages. And this is what leads to a diversity of migration patterns. It leads to residence and migratory early and late seasonal migrations. But what the key thing is here is that a group of animals have different migration patterns that lead to a diversity of migration patterns. So I'm going to start now with a study led by Gaddy et al., published in ICES. And what they did is they had a lot of PSATs, or pop-up satellite tags. They had 62 PSATs that provided completely archived data sets with high temporal resolution. And they had 4 years of data from 2013, 2014, 2015, and 2017. And they tracked fish from different sites to a common channel where they detected spawning in the winter and returned to their home sites in the summer. And so that was known as, or what they have done is they have found summer site fidelity in these halibut in the Gulf of St. Lawrence. And interestingly, the variation in the distance traveled, and it was related to availability of spawning habitat and the size of the fish. So in terms of the availability of spawning habitat, you can see these fish up here, they didn't have to travel far to find a good habitat. Overall, the distances that were traveled ranged from 3 to 324 kilometers, and the median was 27 kilometers. So they are not, they're migrating off, but coming back to their home site. On the Scotian Shelf right now, draw your attention, a study was done by Armsworthy, Trzynski, and Campana that looked at animals on the Grand Banks, and 2, I guess, or 3 on the Scotian Shelf. They also, there were only 14 of them. We're sort of updating that now. They looked at 14 of the fish, and they found a seasonal pattern that was consistent with what they found in the Gulf of St. Lawrence, which was that they winter at depth on the slope, and they move towards the shelf in the summer. Again, summer site fidelity. They move to the same site. So the distance, in terms of the distance traveled, the most fish did not stray more than 120 kilometers from their tagged location, and 4 only moved, they moved less than 25 kilometers. And now we're going to move on. Atlantic halibut in the Gulf of Maine based on geolocation. This is a study led by Chang Liu. And what you can see is that in this particular instance, of 35 fish, 80% remained within the Gulf of Maine, and further, they only traveled less than 60K, and 1 got eaten, and it's described in the paper if you're interested. So that was cool. So what happens here in the Gulf of Maine is that the majority showed little movement. in fact, most of them stayed where they were, although 2 made putative spawning runs from a different home site to a common channel. And that is shown here. So here are the 2 fish that actually did travel and overwintered in this area in the Fundian Channel, but they came from different locations. They returned to these locations, but they spawned here. And so if you, each of all of the triangle and the circle are color-coded by the month of year, and you can see that there is some overlap in those areas. And so this is really of interest, and it does almost tell us that this may be a place where they mix, but contingents are acting differently. So the contingent theory. So to sum this up, what we have is contingents within a population develop migration pathways as a result of environmental cues. We see seasonal migratory individuals and groups. We also observed residents. We're not sure why. There could be skipped spawning. And also emigrants, and these would be the explorers encouraged by crowding. The bottom line here is that what we end up with is a diversity of migration patterns. So that's all the pop-up, tagging from electronic tagging, but we also have all the conventional tagging as well. And Krista Rancière, an author on the paper as well, she put that all together and produced this plot, which takes many studies and puts them on one plot. And to walk you through that, we're going to first focus on the median distance recapture from conventional tagging, which is in the circles. And so as an example here, these animals, they're median distant at recapture. They were tagged with conventional tags. They were released. They were caught some distance later and returned to fisheries and oceans or to the study. These ones traveled 16. These ones traveled, median distance was 9, 33, 3, 32, 11, 56, 89, and 38. So these are all small compared to the unit stock, or compared to the size of the stock structure. We also, as a recap on this, we also put together the electronic tagging, which shows potential spawning sites, as well as summer feeding grounds and winter feeding grounds, both in the Gulf of St. Lawrence, as well as the Gulf of Maine. And we're hoping to fill in this area soon with the Scotian Shelf information. So as a result of that, we have also all the conventional tags, which really do tell us that we have to pay attention to these migration patterns in terms of the management. I'm going to go to nursery areas and connectivity. I should probably check the time. I'm good. Nursery areas and connectivity. This is actually work done by Stephanie Boudreau, who was also at Dalhousie. She did her PhD at Dalhousie, who's currently at Fisheries and Oceans in Moncton. And what she did was ask the question about connectivity across a landscape of juvenile halibut. She used a spatial temporal model, and that Bayesian hierarchical spatial temporal model has 3 parameters that can have strong application to spatial management: connectivity, spatial variance, and persistence. And I have the star here because this is showing you that this is one of the better statistical approaches that we can apply to manage fish better. So what are connectivity, spatial variance, and persistence? This is a depiction or a schematic showing those 3 parameters, which you can extract from the model. Persistence describes the spatial pattern, and if they are highly persistent, it means that they are the same year after year. So I've got these 3 red dots, and we always find fish in these locations year after year after year. And where there are no red dots, we never find fish. So what it's saying is this spatial pattern is the same year after year after year over time. The connectivity tells us how those animals or how the temporal abundance changes over space. So here we see these animals are increasing over time. They're doing exactly the same thing, following my cursor. They're doing exactly the same thing on the Scotian Shelf as they are in Newfoundland. So these animals are highly connected, or the connectivity is high. In contrast, there's low connectivity if the temporal patterns are not the same at all. They're all crazy and doing different things in different areas. That means this is low connectivity and high connectivity. And spatial variance is really the difference between the hot and cold spots, and really is almost like a vertical, you know, how the difference between a low variance is kind of evenly spread, and a high variance, they're large patches. Today, we're only going to look at persistence and connectivity, and that is here. We now have maps of juvenile halibut in 3 time periods: 1978 to 89, 1990 to 2003, and 2004 to 2013. And what you can see here is that there are persistent patterns over time. They're always in the gully. We also saw this with the fishing landings. They're always in the gully, and they're always in Southwest Nova. They're always in the Gulf of St. Lawrence in the same spots. Those areas start to grow, except southern Newfoundland starts to disappear. So what we've found here is these are independent, persistent areas of high juvenile halibut abundance. Those are always there, relatively more dense than other areas. So we would call these core areas. So how are those core areas connected? If we look at the model, we can see that those core areas, they have coherent temporal patterns, meaning that they sort of increase and decrease at the same time over time, at a distance of, on average, of 250K. So 250 kilometers is roughly here to here. These animals are roughly acting the same, and but 250 kilometers is much smaller on the Scotian Shelf, much smaller than the current domain that we presume. As you'll recall, we presume that there's one stock from southern Newfoundland all the way down to Southwest Nova. That's roughly 2,000 kilometers, but the temporal patterns are operating on average 250 kilometers. We then also looked at habitat suitability. We wanted to know if nursery size was related to adult production. So the habitat suitability map is on the top, but I'll just draw your eyes down to the bottom 2 graphs. So what we have is the suitable habitat share among all these NAFO divisions. So we're looking at, like, for example, Forax has most of the lion's share of halibut habitat. And we've contrasted that with landing share in 2014. So more fish, the greater share of fish were caught in Forax, but they also had most of the habitat, juvenile habitat. And you can see that there's quite a nice line that goes down in terms of where you have a lot of juvenile habitat, you're going to get a lot of landings. And this really speaks to something that is called the nursery size hypothesis, which says that adult production is related to, in flatfish, is related to the size of the nursery. So this conforms to that theory, and it conforms now, and interestingly, it conforms to landings back in 1953-54. So we took those landings from 1953-54, and of course the lion's share of those landings way back in 53-54, before most of us were born, came out of Forex, where we have the most suitable habitat. It also suggests local retention. So that's it in terms of nursery areas and connectivity. To sum up, we found core areas, and we also can estimate the connectivity between them. And we know that larger nursery areas contribute to a higher adult production. So now we'll move on to growth and maturity. And growth and maturity, or life history traits, just as an introduction, this is from Armsworthy and Campana. And halibut are large, long-lived, and sexually dimorphic. So here we've got age on the bottom and length on the y-axis, and you can see that females on average are larger than males. So males can mature at a smaller size, and maturity is, when this growth rate starts leveling off. But what you can also see is that there's a lot of variation around this line. If we focus in on the females, we can see that there's a huge amount of variation, roughly at 10 years. So a 10-year-old female could be anywhere from 75 to 175. We wanted to look at that variation more because, as you'll recall, we know that there's a gradient going from northeast to southwest, and we wanted to look at the influence of temperature and growing degree days. Maybe it's contributing to this variation. You'll also recall that part of this variation is because the stock is managed or estimated over such a large area, all this area in purple, teal, this bluish color. You can imagine that that variation comes from just doing this over a very wide space. So we did that. We used whatever growth rate from tagging, and some of it was observer data. So I'll start first with growth rate in females and males. We used actually a model using growing degree days. We used it to predict the growth rate from tagging data. And what we can see is females are pink and males are blue. And here we have 4x to 3n. So this goes from south to north roughly. So 4x to 3n. So we're looking at this gradient very similar to this map. And we can see that females and males grow faster in the south than they do in the north. Males as well, they grow faster in the south than they do in the north. When we get to length at maturity, what is the length of maturity? We do see a pattern, but we were using observer data, which was really hard. So when we came to calculate the credible intervals, we actually included the observer random effect variance, just to be honest about it, and to use this as a starting point. And even when we do that, we can say females, which are again pink, in 4x, there's a higher probability of them maturing at a smaller size than in 3n. Males, it's a little bit more messy, but certainly for females, we can say with in all likelihood, a female in the southern part of the range is going to mature at a smaller size than a female at the northern or northeast part of the range. Okay, so in summary, we've got all these updates on Atlantic halibut spatial stock structure. I'm going to start with the genetics. We have subtle but significant differences between the Gulf of St. Lawrence and the continental shelf. We did not find a difference between the shelf and Georges Bank and Gulf of Maine area, which means we really do have to figure out, that's an area of current research to focus in on that area, because this means either that they straddle the border. We do have to worry about overfishing one side, or is the US fishing our fish? Are we fishing their fish? We go on to migration and all the work coming out of the Gulf and Maine shows that we now understand that they winter and spawn on common slopes, and they return to their home foraging site for summer. And that is related to variation in timing, size, state, and distance to slope. In terms of nursery areas, we see these persistent juvenile core areas that are separated by 250 kilometers, which is much less than how we currently manage those stock now, and that the size of the nursery area is related to the landings. Then finally, we found that southern fish grow faster and mature at a smaller size than do northern fish. And this is consistent with many studies that have shown similar patterns in warmer water. So the warmer water speeds up your growth rate and your and you reach a size at maturity earlier. So finally, we have it all summed up, at least the migration, and this is a schematic lifestyle of Atlantic halibut by Ed Beals. And so the juvenile hotspots are in yellow, and those that are very solid show where they are. And then as they diffuse, this means that they start spreading out, the distribution starts spreading out. The spawning areas are in the deeper waters. As they mature, they come down. The seasonal migrations as they come down, but we do know from conventional tagging data and some of the electronic tagging is that sometimes some animals will go. Is it enough to swamp genetic differences? Maybe, maybe not, but these animals are large, and if we can imagine that if there are too many in one spot, they may leave. So the final remarks, we've a whole bunch of people are interested in Atlantic halibut, especially under climate change. We know that the need for basic ecological research is greater, and I think we've learned from previous studies that we need to understand the mechanisms so we can anticipate the response to changes in the drivers. This is really important knowledge for managing in a dynamic environment where we don't know. We have some ideas about spatially structured models. Brad Hubley does. Lingbo Li is on the case. All of that's being worked out now. There are many questions that still persist, but we can proceed with the information we have now. Both the spatial and economic scale of this species are raising new ecological and management questions, especially across the border in the eastern Gulf of Maine, how we are going to sort of resolve that particular issue. And we also have all these promising new approaches to spatial stock assessments, as well as management strategy evaluations. So we are well equipped to manage this halibut sustainably. And lastly, the questions are increasingly suited to regional and international collaborations. And this is a contribution, this last bullet is from John Fisher, who believes that, you know, this large raging fish across the Atlantic region, we've all benefited a lot from working with each other and sharing information. And that might be it.